let's start. So I think you are a botany honors student and you are in the third year. Am I right?
Yeah, yeah. Actually, so there are the students from three
Second year,
semesters.
fourth year, and sixth year. Means
Okay.
6 sem actually.
Sixth sem and
Second sem, fourth sem, and sixth sem.
okay. Three batches are there. Okay. So let's start. This is under your sixth semester syllabus. It is under plant biotechnology and genetic engineering. So in the unit 2, it is written that in your syllabus that a restriction endonuclease. So just I today I try to give some concept on this particular enzyme. Actually, I think that you have the a sound knowledge on DNA structure. DNA is a double-stranded helical structure, and these two strands are joined by hydrogen bonds, and these hydrogen bonds are made between A and T and between G and C. And just let me know whether this what I have said now this is known to you or you didn't know that. Anybody, please? DNA is a double-stranded. It is a helical, and
For the sixth sem student, they already knows it actually. And for the
uh,
second and the fourth, they are new to it,
oh,
I guess.
they don't know regarding this DNA structure even, is it?
Ah, in their 11, 12, they have all read it,
Okay,
I guess so.
okay,
Right.
okay, okay. So in the DNA double-stranded, and these two strands, if I consider any single strand, they are made like hmm, so pictures. So if you consider this is one single strand and this is another strand. So if I consider any single strand, uh, they are made by four nucleotide A, T, G, C. So suppose the sequence is G, A, T, T, C, what is written here, and in another strand it must have its complementary uh, nucleotides. So suppose in one strand if it is G, so in opposite strand it must have C, because G can only make hydrogen bond with C, and if it is a, then in opposite strand it must have T, because A can make hydrogen bond only T. So by this way, this sequence, that G A A T T C, this is just 6 nucleotide base sequence, it is written here, but in your body, in each of your cell containing near about some 10 to the power 9 numbers of nucleotides, long chain. So actually this sequence varies from person to person, from organism to organism, and actually particular these millions of this particular sequence actually make a particular individual. So if this is true, and if this strand make hydrogen bond with another strand which is complementary, these two strands actually the concept of the double stranded helical structure of the DNA. Now if I just, since they are added bind with the hydrogen bonds and that is a very weak bond, so if I apply little bit heat, then these hydrogen bonds will break and two strands will be separated. But in the other way, if I want to break the single strand, that means this GAATTC, if I want to break this anywhere from this single strand, it is very much difficult, because this G and this adjacent A and its adjacent another A, and like this way, they are bound with the phosphate bond. So one here having one sugar. This sugar can make one phosphate bond with the next sugar of the next nucleotide, and by this way sugar phosphate sugar phosphate sugar phosphate bonds are there, and from the each sugar this one base that either G or T or C or A are attached. So by this way one single strands are produced and its complementary strands are joined by the hydrogen bond. So again I am telling that this double stranded DNA easily I can make it single stranded by simply applying heat. But to cut the single stranded DNA, it is not so easy. If you apply heat or if you just char it, burn it, it will not cut the single stranded DNA. Now coming to the first slide, recombinant DNA technology. So in your syllabus, that is genetic engineering, so here in this technology we will cut, we have to cut the DNA, we have to change the DNA sequence, we have to modify it, and we have to incorporate it into a particular host body to get the certain effect or advantages. Like if I want to say that today we are getting human insulin from medicine shop. So person suffering from diabetes, they need insulin, but from where we will get the insulin? We will get the insulin either initially it was from the animal insulin, but there was certain problem, but the human insulin not easy to get, because for that we have to kill the human and collect the pancreas and then collect the islets of Langerhans cell, and then we need that insulin, but which is not possible. So instead of that, if I can collect the insulin gene of a normal individual who can produce normal insulin. So normal insulin is a protein. This protein is produced from one particular gene. Gene means one particular part of the DNA which can produce that protein. So if I can collect that gene and that gene can cut it, we can cut it if, and this particular insulin gene, if I can incorporate into other host cell, that may be one animal, maybe one plant, or even it may be one bacteria, and if that particular organism, that plant, animal or bacteria, can produce that insulin gene, that means indirectly we are producing that human insulin, because that gene is from one human, it is just incorporated inside the other species. Now for that, first step is we have to cut that insulin gene from one normal individual. How to cut? So this particular scissor which can cut the particular DNA in a particular location, that is called restriction endonuclease. And this is the primary tool for the recombinant DNA technology or biotechnology or your genetic engineering. Because without cutting we cannot modify the gene, or without modifying we cannot go for the, we cannot change the genetic character. Now the steps involved in the RDNA technology is, first is identifying gene. Identifying gene means in your body, if it's in a human body, in each cell there are about nearly 32,000 genes. Out of this 32,000 genes, if I want to collect one particular one, for example insulin gene, so this is first step is identifying. Otherwise I have to identify, for this instance, insulin gene. Now after identifying, I have to cut that gene. In your chromosome there are several genes are located. How many genes? Near about 32,000 genes. Out of 32,000, I have to cut particular one, that is, for example, insulin gene. This is isolating gene, and this is done by restriction endonuclease. Then the steps involved is that isolated gene, which is insulin gene, is uh, placed into the suitable vector, and that vector is placed into a suitable host cell, and there it is re-expressed. That means that gene will produce that corresponding protein. For instance, insulin protein will be produced in that particular host cell. So we will concentrate this particular one, this isolating gene. Hey, শোনা যাচ্ছে আমাকে?
Yes, sir. Yes, sir.
হ্যাঁ স্যার, হ্যাঁ স্যার, শোনা যাচ্ছে।
Okay. Chalo, restriction endonuclease. Who produced that? Bacteria produced that. So bacteria is a eukaryotes which can produce this particular restriction endonuclease. They produce to protect themselves for virus attack, and the virus which can infect the bacteria is known as bacteriophage or simply phage. So they enter inside the bacteria body. Actually, bacteriophage generally not enter inside the bacteria. Rather, bacteriophage first attach with the bacteria, then it inject its genetic material to the bacteria, and that genetic material, that means the DNA and sometimes RNA, actually take control of the host cell. Here host cell is bacteria, and then it dominate there, ultimately kill the bacteria, and they produce that genetic material of the phage, produce huge amount of newly produced virus, that means phage. They come out after lysis the cell membrane of the bacteria, they come out and they infect the surrounding cells. So ultimately they kill the bacteria and can infect the surrounding cells, and surrounding cells also will be killed by this newly produced virus, that means bacteriophage. So this process is going on, and if it was allowed, then bacteria could not survive. So bacteria have its own immune system. What is this? Bacteria, after injecting this genetic material, this phage genetic material, when it is incorporated inside the bacteria, then bacteria produce restriction endonuclease, which actually cut the DNA or genetic material of this phage, so that phage cannot do further attack. So phage will be killed here, or the phage genetic material will be inactivated with the help of restriction endonuclease. It was the story. So this war between the virus and the bacteria is going on from long ago, some millions years ago, but we discovered that mainly in 1968 and 1970, this existence of this restriction endonuclease, mainly 1970 we got it, and we came to know that this enzyme having a huge power to cut the DNA, and we can modify our DNA according to our will with the help of this restriction endonuclease. This name implies restriction because it restricts the attack of phage to the bacteria. That's why it is known as restriction. And in other way, it is known that not everywhere that enzyme cut the DNA. It can cut within a very much specific location. So since it is cannot cut in the elsewhere, so it is known as restriction. And it is endonuclease. You know, nuclease. So nuclease, if it is AACase, means it is an enzyme. Nuclease means which can degrade the nucleic acid. So that's why it is nuclease. Any enzyme which can degrade the nucleic acid, DNA or RNA, it is known as nuclease. And if it cut within the DNA molecule, that is known as endonuclease. So another term, the opposite term of endonuclease is exonuclease, where this enzyme can cut from the end. but it is cannot cut from the end, it can cut from within the sequence, that is endonuclease. So total name is restriction endonuclease. In short, it is known as RE, capital R, capital E, or simply it is known as restriction enzyme. Now suppose this is viral genome, it is entered inside a bacteria, and it is a long gene. Suppose some thousands of nucleotides are there. So this sequence GAATTC, before that in the left side there are thousands of sequences, and the right side there are thousands of sequences. So if a particular, out of these some thousands of sequences, if there is a particular sequence, that is 6 BP sequence GAATTC, then only one particular restriction endonuclease. I told you that restriction endonuclease is produced by the bacteria, and different types of restriction endonucleases are produced by the different types of the bacteria. One particular bacteria, for example E. coli, Escherichia coli, you have heard the name definitely. So this bacteria produce one particular restriction endonuclease which is known as EcoR1. So that not means that E. coli only produce EcoR1. It can produce something other, other restriction endonuclease also. But if it is EcoR1, for instance, then it can recognize this particular sequence. Which sequence? GAATTC. If it can recognize, if this particular sequence is present in the viral genome, then this particular enzyme bind here. After binding, this enzyme can cut between G and A in both the strand. So if upper strand, there is a two strands, you can see in upper strand 5 prime GAATTC, and in lower strand, if you read in opposite direction, here also 5 prime GAATTC, same thing. Upper strand if you read through left to right, and lower strand if you read right to left, you will get the same thing. Now in upper strand, if it cut in between G and A, and in lower strand it will also cut in between G and A, then this DNA molecule, single DNA molecule, one DNA molecule will be fragmented and will give two fragments. Now it is two DNA molecule. So one is this part, left part, some thousands of nucleotide may present, ends with G because it cuts here, G, and in the lower strand, these thousands of nucleotide ends with CTTAA, that means end with A, because here it cut between A and G, so it will proceed up to this A. So this is about one fragment, left fragment, and for this one, I think you can understand that the upper strand starts from A due to this, and lower strand starts from G. Do you get it? বুঝতে পারছো? না স্যার, বুঝতে পারছি না। আমি আছি তো, না বেরিয়ে গেছি? হ্যালো,
হ্যাঁ শুনতে পাচ্ছি স্যার।
ওকে, না আমি তো বুঝতে পারছি না তোমরা আছো কিনা বা আমি আছি কিনা। চলো, তো so by this way the DNA fragments are produced, and if bacteria itself have the same sequence, then its own produced restriction endonuclease cannot cut it, because it's that particular sequence is methylated, it is modified mainly A region, adenine region, and C region, cytosine region are methylated. CH3 group is attached here, so that its own restriction endonuclease cannot bind here, and since it cannot bind, it cannot cut. So bacteria own DNA is protected by methylation, but since viral DNA is not methylated, so it is affected by its host cell RE. RE means restriction endonuclease. So this is the mechanism. Now there are different types of RE as per your syllabus, and that is type 1, type 2, and type 3. And in your syllabus there are also type 4, but which is generally there are little application in recombinant DNA technology. Now out of this type 1, 2, and 3, which are written in the Roman number 1, 2, 3, type 1 and type 3 are having some specific common feature, and type 2 having some different character. So if I keep type 1 and 3 together and type 2 separate, then this 1 and 3 cleave the DNA non-specifically. Cleave the DNA non-specifically means they bind, they recognize a specific sequence. This is type 1. They recognize a specific sequence like GAATTC, what I have said, but they cut somewhere else, not within that sequence. So that cut or cleave is non-specifically. This is also true for type 3. It can recognize one specific sequence, can bind with that specific sequence, but cut somewhere else, not within this sequence. But only type 2 can recognize a specific sequence and also cut within the sequence. So if I again come into this picture, this is the example of type 2, because it can recognize one specific sequence, which is GAATTC, and cut within that sequence specifically. Here it is in between G and A, but it is not true for 1 and 3. They will bind with the specific sequence, but they will cut somewhere here or somewhere here, generally not within this sequence. So cut is not specific. Again, all these three can recognize the specific sequence, but only type 2 can cut specific region, and type 1 and 3 cut non-specifically. So these are the basic difference between these types. Again, type 1 and type 3 having bigger size and having lots of subunits which are not identical. So in upper stand, one subunits can bind, in other lower stand another subunits can bind. So in upper stand it can cut somewhere else, in lower stand it can cut somewhere else. So this problem is there for type 1 and 3. Since they are bigger, they can cut some distance from the recognition site. But type 2, which is small and having only 2 identical subunits, one subunit bind with the upper stand, another subunit bind with the lower stand. So since they are identical, so if upper stand cuts in between G and A, lower stand will also cut in between G and A. So actually in RDNA technology or in your genetic engineering, this type 2 is widely used because it can cut specifically. Type 1 and 3 require lots of cofactor, and type 2 only required magnesium as a cofactor. So these are the basic difference between these two. So type 2 is actually used. So they actually recognize the palindromic sequence. What do you mean by palindrome? I think you know, or some of you know. Palindrome means if you read any word or letter from left to right and right to left, you will get the same answer. So for example, these are the palindrome. So from left to right or right to left, just you go through, you will get the same answer. So type 2 actually recognize this palindrome sequence. So pictures are not. So palindrome, for example, this is echo R1 as it was shown initially. GAATTC, echo R1 recognize this, and if you go through that lower strand and just opposite direction, then you will get the same thing, GAATTC. And for another restriction engine, for example, name is Hind3, it can recognize AA 5 prime AAGCTT, and in lower stand also, if you go through, you will get 5 prime AAGCTT. So these are palindrome. In case of DNA palindrome, uh, please don't go through the same strand. If you in a single direction, if you go through a upper strand, just for lower strand you go through the opposite direction, then only you will get the same answer. If it is true, the name of the restriction endonuclease, that means the nomenclature that I have said, that echo R1, HIN3, this type of names, how they are named. So the convention is the bacteria which is produced that enzyme, the first letter is the first letter of the genus name. So if it is echo R1, for example, echo R1, this E from the first letter of the genus, which is Escherichia, and it is capital. And next to is the species name, small letter, and the first two letter of the species name, here it is from coli. So E for Escherichia, CO for coli, and Escherichia coli can produce, sorry, Escherichia coli having different strain. So what is strain? I am not going into the details of that. So particular one strain which is named as R, capital, it is written capital. So particular one E. coli strain which is known as echo R. This echo R can produce more than one types of enzyme. So to distinguish them from one to another, since all are produced from the same strain echo R, they are designated as Roman 1, 2, 3, 4 as per their invention. So one was invented first, and then laterally 2, 3, 4, 5. So there have echo R1, echo R2, echo R3, even echo R5 are there as per their invention. If I ask that Haemophilus influenzae can produce one restriction endonuclease, Haemophilus influenzae strain D produce one RE. So what will be the name of that RE? Anybody? Nobody? The genus name is Haemophilus. So the first letter is H, capital. Next is influenzae. The first two species letter is influenzae IN. That is HIN, and the strain I told you that is D. So it is HIND, Hind. So this is Hind3. That means Haemophilus influenzae strain D, and this can produce different types of RE, according to their inventor, they are denoted as 1, 2, 3. So this is particular one, that is hin 3, which can recognize this particular sequence. So there are hundreds of RE are available, and they can recognize the different sequences. So this is your nomenclature part. So it is described that if there is a sequence GAATTC, then EQR1 will bind and two fragments will be produced, and the end of the each fragments will be this and this. Now just look carefully that in left side here there are thousands of nucleotides, in right side there may have thousands of nucleotides. So here also after cutting, this is thousands and thousands, and it ends at G and it ends at CTTAA. So this, if you look carefully, that TTAA, this part is single stranded part. It's don't have any double stranded region. Up to this G can make hydrogen bond with C, but this TTAA is remain alone. And same thing for this opposite fragment, another fragment. These have the single stranded part, which is AATT, not C, because C is already having pair with the G. So this single stranded part AATT, and here also the single stranded part TTAA exists. So irrespective of that, the sequence of here, this left part, or here this right part, if it is treated with EQR1 and any DNA molecule, and if it is fragmented, then whatever may be the sequence of here or here, the end sequence is now known to me, and that is this TTAA and AATT. And another strange thing is that if this molecule and if this molecule are again kept together, then this A can make hydrogen bond with this T, that I told you that A can make hydrogen bond with this T. This T can make hydrogen bond with A, and this T with this A, and this A with this T. So they will again join, but this G and A, this gap will be there, and this A and G, this gap will be there, because this was made by the phosphate bond. So except this phosphate bond, they will make this hydrogen bond and they will join again. So what I want to say, that if this fragment and this fragment in a test tube, if we keep and allow them to join, they will join, because their single stranded parts are complementary to each other, and they will again join. And if I add in that test tube DNA ligase, that is one glue atha type of enzyme which can provide this phosphate bond, then this G and A will be joined, and this A and G will be joined. So soja kotha, this eco R1, if I treat here in a long DNA molecule which have this particular region, then they will be separated and make two fragments. It is true. And again, if I add in that test tube some DNA ligase and allow them to join, then these two fragments again produce this one single molecule. Do you get it? I think this is the problem, because I cannot see your face, so I cannot understand whether you get it or not. So this single stranded part which is extended remain is known as sticky end. Sticky, S-T-I-C-K, sticky end, athalo. Because why it is sticky? Because if it gets its complementary region, then again they will join. This is also true for KPN1. This is KPN1 can recognize another sequence, for example 5' GGTACC, and it can cut in between C and C. In upper stand in between C and C, in lower stand it is also in between C and C, and these fragments, two fragments will be produced. Here also the sticky ends here is GTAC. Here also the sticky ends are CATG, which are complementary to each other, and they will be joined if we allow them. But in case of HEPA1, it will cut in between T and A, since this is 6 nucleotide long, and if I cut between third and fourth, that means just in the middle, then it will give that T here, last one is T, and here A is last, so it's don't have any single stranded extended part, and since it's don't have any single stranded part, it cannot join with the other DNA molecule because it don't have any complementary region, and that's why it is known as blunt end, BLUNT, blunt end, bhota. So blunt end is a little bit difficult to join them again, but they are naturally, if you keep together, this and this will be joined if you add DNA ligase. So this is your eco R1 having two identical subunit, because it is type 2, so two identical subunit, and it binds, one subunit binds with the upper and another one is lower, and they cut in the same precise location. Now students, let me know, they are the some example of the RE. Here only single strand is shown, because if you get the single strand, you automatically can get the another complementary strand. So eco R1, if I say that it can recognize 5 prime GAATTC, so I think what is the lower strand, another strand, you can get it. Just this G having complementary with C, this A having T, A having T, T having A, T having A, and C having G. So if you go through this opposite direction, you will get same thing, G, A, A, T, T, C, and it will cut in between G and A. Now, eco R5 can cut GAT, ATC like that. So single standard things are shown here, another standard is not shown. You tell me, after treating this RE, who can make sticky ends? Just you give the answer in yes or no. Who will make sticky end? Eco R1, will it make sticky ends? No answer. ইকো R1 স্টিকি এন্ড তৈরি করবে? ইকো R1 দিয়ে যদি আমি কাটি একটা ডিএনএ মলিকিউলকে, কেউ বলো বাবা, আমি তো বুঝতে পারছি না।
এর টিটিসি এন্ডটা স্টিকি এন্ড ক্রিয়েট করবে।
So it will make sticky end. Okay, very good. Eco R5, will it make sticky end? No answer. Hint 3, sticky end তৈরি করবে? বলো বলো।
Yes, sir.
করবে, ভেরি গুড। SAC1 করবে? বলো, কেউ বলো, জলদি বলো, কুইক। Yes sir. Okay, let me know which one will not create sticky end, which one will make blunt end. Anybody please? Blunt end. Eco R5
Eco R5.
will make, very good, Eco R5 will make blunt end. Another, any other enzyme?
SMA1.
Very good. SMA1
SMA1.
very good. SMA1 will make blunt end. So out of this, all this, only these two will make blunt end, Eco R5 and SMA1. All others can make sticky ends, because if it cut in the middle part, exactly in the middle, then only it will produce.
So you are not audible.
আমাকে শোনা যাচ্ছে না?
হ্যাঁ হ্যাঁ হ্যাঁ, এবার শোনা যাচ্ছে, মাঝখানে চলে গেছে।
ওকে। Oh, so so this is if it cut just in the middle, exactly middle, that means on the axis of symmetry, then it will produce blunt end. Otherwise, anywhere else if it cut, it will produce sticky end. Let me see which one will produce blunt end. বলো, ব্লান্ট এন্ড কে তৈরি করবে?
Sir, first.
ফার্স্ট এডটা করবে আলু 1। নেক্সট আর কিছু?
হ্যাঁ স্যার, সেকেন্ড এডটা।
ভেরি গুড, সেকেন্ড এডটা। বললে বিশ্বাস করবে না, থার্ড, ফোর্থ এন্ড ফিফথ উইল নট প্রডিউস blunt end, rather they will produce sticky end. I think you will get it. Now coming to the main thing, I will take maximum 5 minutes of time. Now this is your DNA and this is bacteria DNA, suppose. And since your DNA is very long, I told you it is near about 3 into 10 to the power 9 nucleotides. So there are lots of particular this sequence GAATTC, and that is also true for that particular bacteria. For example, this is equally having this particular sequence. Now in a test tube, you take your DNA, and in another test tube take that equally DNA. So these sequences are entirely different. Left, right sequences, that millions of sequences are there. So you will not get any similarities between this. Only there is a one GAATTC which is common in both the DNA. If it is true, then if they are kept in the different test tubes, and in both the test tubes, if I add EcoR1 which can recognize this and will cut this, so it will produce one fragment this and another fragment what will be produced, that you know. And these two fragments, out of two fragments, if I keep the left one and discard Feledi in right fragment in a test tube, and in another test tube the fragments which are produced, if I keep the right one and discard the left one, and then if I mix the content of these two test tubes, that means these test tubes and these test tubes, then you tell me whether they will join again or not. Because this part which is sticky end, extended single stranded part, and this part, since both are produced by the same enzyme EcoR1, so their end sequences, the single stranded sequences, are complementary to each other. As I told you that two fragments may join if you allow. So here also the same thing, if you join, mix these two content, then two fragments will be joined. But I also told you that this gap between G and A, which contains one phosphate, and this gap between G and A, which contains one phosphate, is missing here. They can make hydrogen bond. This A will with T, this A will T, like this they will make hydrogen bond. But between this G and A there the phosphate bond is missing, so it is not a stable one. So in the test tube, what should I add? DNA ligase. As I told you, it is a glue like Fevikwik, so it will make or provide this phosphate bond between G and A, and now I will get one DNA molecule from two DNA molecule. One is obtained from your DNA, another is obtained from equally DNA. and in this single DNA molecule having both the character, this is one sequence from you, this is another sequence from the bacteria, so both the characters are present in a single DNA molecule, which is actually known as recombinant DNA, which contains both your character and bacteria character. So this is the concept of the rDNA technology, and without having this restriction endonuclease, you can't go for anything for this rDNA technology. So this is the basic tool of the uh, rDNA technology, which is restriction endonuclease. Got it? So I think I should stop here. Lots of things are there in your restriction endonuclease. Another type is in your syllabus, that is type 4. I am not going into the details regarding type 4 because only type 2 is used in the rDNA technology. Bolo, if you have any question, please.
Yeah, thank you so much, sir. মানে খুব লুসিড ওয়েতে বুঝিয়েছেন। আমাদের 6 সেমিস্টারের স্টুডেন্ট আছে এবং মানে সেকেন্ড আর ফোর্থও রয়েছে। তো হ্যাঁ, ওদের কাছে একটু নতুন হলেও বাট পরবর্তীকালে নেক্সট সেমিস্টারে গিয়েই ওরা হয়তো পড়বে এই জিনিসগুলো। তো কিছু একটা মানে একটা আইডিয়া হয়ে থাকলো যে কি পড়তে চলেছে এরপরে।
অ্যাকচুয়ালি ডিএনএ মলিকুলার স্ট্রাকচারটা না জানলে এগুলো একটু
হ্যাঁ, ইট উইল বি টাফ ফর দেম। কনসেপ্টটা পাওয়াটা একটু মুশকিল। তো কারোর যদি বেসিক যদি কিছু প্রশ্ন থাকে স্যারের কাছে করতে পারো। ইফ দেয়ার ইজ এনি কোশ্চেন, খুব বেসিকও কোনো অসুবিধা নেই। ডিএনএ কি সেটাও জিজ্ঞেস করতে পারো, সেটাতে কোনো আপত্তি নেই।
হ্যাঁ, কোনো অসুবিধা নেই। তোমরা জানো না, আমাদের কাজ তোমাদের জানানো, এটা নিয়ে
জানানো, হ্যাঁ
চাপাই মানে এর কিছু নেই। অ্যাকচুয়ালি সবার 2টো বাজে তো সবার খিদে পেয়ে গেছে আই থিঙ্ক।
হ্যাঁ, সকালে আরেকটা ক্লাস ছিল ওদের অ্যাকচুয়ালি, প্রায় ওটা 1.5 ঘন্টার মতন চলেছে। তারপরে আপনার ক্লাসটা ছিল আজকে। যাই হোক, যদি ফিউচারে কোনো প্রশ্ন থাকে ইউ ক্যান কন্ট্যাক্ট মি, আই উইল ডাইরেক্ট ইউ টু দ্য স্যার। সো থ্যাংক ইউ সো মাচ স্যার আজকে আমাদের একটু সময় দেওয়ার জন্য। ওকে, থ্যাংক ইউ স্টুডেন্ট। তো আমরা এখন লিভ করতে পারি তাহলে।